So as God said, there will be a lot of overlap of my talk with Joe's talk previously this morning. I'll talk about some findings that we have from the exome sequencing we do at the Autism Sequencing Consortium, and some of the findings we see in the Israeli population. Okay, so this slide shows you, what you already know, that there is no doubt that genetics plays a significant role in autism etiology. Many studies estimate the heritability of autism around somewhere between 40% to 90%. It means that genetics is a major player in autism, but still identifying the genes or the genetic factors underlying autism is very, very difficult. This is probably because what you see here, the genetics of autism is very, very complex. There is a huge variation when we are looking at genetic variation underlying autism or causing autism. There is a huge variation both in their inheritance patterns, as you can see here, both recessive inheritance, X-linked inheritance, de novo mutation, as you heard from Joe earlier. Everything is associated with autism. In terms of the type of variation, we see both translocations, single nucleotide variations, copy number variation. Again, almost everything we know from genetics exists in autistic patients, and so also a huge variation in the penetrance, from the highly penetrant variation, again what you heard from Joe earlier, to the very low penetrance, usually common variation. Also, we can find in autistic patients. So genetic studies taught us that autism probably is not a single disorder. It's more like, so we refer to autism like currently as a spectrum, but it's probably not a spectrum. It's possibly more like a puzzle, which each individual carry a different variation, a different gene maybe, that leads to different manifestation of autism in this individual. And this is probably why it's so difficult to identify the genetic factors of autism. So this actually leads us to two major challenges in autism genetics. The first one is to identify the genetic causes of that in different individuals with autism. And this is actually currently partially resolved by exome sequencing, as you heard earlier. Exome sequencing has been proved to be a very efficient approach to identify mostly the rare variants of autism. But as Joe already showed you earlier, with large sample size, we can also identify common variants with low effect. We can also maybe use this sequencing data to identify copy number variation, but this is more complicated. But this is currently the most efficient way to identify new variant associated with autism. The second challenge is to understand once we find the variant, what we think is the underlying variant in each individual, to understand what's the phenotypic manifestations that are associated with specific variants in autism. And this actually require an interdisciplinary approach. And this is actually the reason that we established what is now called the National Autism Research Center, previously called the Negev Autism Center, 5 years ago at Ben-Gurion University. We started to collect data 5 years ago. We published this paper. We collect about data from around 150 new children every year. Currently, we have data on more than 1,000 individuals in this database. What is important in this database is that every child that comes that with suspicious autism that come to Soroka or all the other clinics that work with us, they undergo the same diagnostics pipeline. So we collect the same data on all of the individuals in our database. They all have ADOS test, IQ test, and finally they receive a diagnosis according to the DSM criteria by a physician. And as you can see in the bottom, they also some of the patients go to different study, participate in different study where we collect additional data like sleeping behavior, sensory sensitivity, we take some biological samples, genetics, and so on. This is some of the characteristics of our cohort. You can see that most of the children are diagnosed very young, under the age of 3, which is also something very unique to our center. We also see the 4-to-1 ratio between males and females, like Florina mentioned before, in our sample. What you can see on the second graph is that around 90% of the children in our center are born in Soroka, so we have access to their complete medical data, including birth data and pregnancy data. And about 25% of the children in our data are of bedding families, which about 60% of them are consanguineous families, meaning they have interfamiliar marriages, which is very important for genetic studies. In terms of genetic studies, what we do, we collect saliva samples from families, from trios or from families, and then send them to Joe's lab for sequencing. They are actually sequenced at the Broad Institute. And so, as we mentioned, we are participated in the Autism Sequencing Consortium. And then we receive back the sequencing. And these sequencings we are analyzing using an in-house bioinformatics pipeline that we developed here. So this pipeline was developed together with the bioinformatics units of Verit Caspi here at Ben Gurion, and one of my students, Apur Rashil, who also helped in developing this pipeline. So this pipeline involves, first of all, we are running this sequencing through the GATK variant calling. Basically, it's looking on all the sequencing and trying to tell where we see a variation that looks real, because there are a lot of false positives in these sequences. So this is the first step in this pipeline. And we consider in this initial analysis only the rare variants, only those with population frequency of less than 5%. The next step, we are running it through the variant effect predictor. Variant effect predictor annotation, meaning that we are trying to understand what is the functional consequence of each of these variants. And again, here we consider only those that are what we call a likely pathogenic variants. Either they are a frame shift that introduces a premature stop codon, or actually causing a stop codon in the middle of the sequence. Those that are either splice donors or splice acceptors, and also missense variant that we are running through two other algorithms called the SIFT and the polyfin that try to predict the functional consequence of this missense variant, and we take only those that are predicted to be deleterious. The next step, we are analyzing the inheritance pattern of these variants by comparing the child sequence or the child variant to his parents. And again, we marked and we consider only those that are de novo variants, X-linked, recessive inheritance, and compound heterozygote, meaning that we take these variants that are unique to the child compared to his parents. Finally, we run another filter for false to identify false positive. Like I said, in an exome sequence, we have a lot of false positives. So we check whether we see more than three de novo variants in our sample, more than three de novo variants in our samples. It means that it's unlikely to see three consecutive de novo variants in the same locus in such small samples that it's likely false positive. And we also, the remaining variants that go through this filter, we also manually curating them using the IGV software. Finally, what we do is we looked only on variants that in autism or non-autism genes, around 200 genes, that we collected from a SFARI database and from the ASC. SFARI database, they give scores in terms of how much they believe in the association of this gene to autism. We take only the top ranked genes in this database, around 168. And also we consider genes that were found by Autism Sequencing Consortium, which got an FDRQ value below 0.05. So what did we find? So currently there are around 770 children in our database. About 1/3 of them, we collected saliva samples and then a sample of around 1/3 of them. Half of these 1/3 were already sequenced and analyzed. So we have exome data for about 121 children. And of these, we identified a likely pathogenic variant in about 20% of these children. The type of variation that we identify, it looks like that. Most of them, as you can imagine, are de novo variants, but some of them are recessive, X-linked, and compound. All of the recessive, X-linked, and compound variation are missense, and also most of the de novo variants. But in the de novo variant, we also see a premature stop codon and some frame shift. Interestingly, we found one child that has 3 de novo variants in 3 different genes, and another 3 children that have 2 affected genes with LPV. And another interesting thing is we found 5 different variants in one gene, and these are 5 different variants. Each individual have a different variant in the same gene. Now I'll show you some examples of the finding or a specific example of the finding that we found. So this is an example of de novo variant in one of the top genes, CHD8, that you can see in the ASC Manhattan plot. It's one of the top ones. We found a de novo insertion that leads to premature stop codon right in the middle of this gene. This is a U gene. I think it's exon number 17 or something. And as you can see, it causes premature. We don't find this mutation in either the father and the mother. And this is de novo. This is a classical finding from exome studies. Another example, very interesting example, another one of the top genes, SYNGAP1. This is actually an interesting story, a Bedouin family where in the first sequencing sample that we sent only the first child with his parents. We found a recessive inheritance, a missense in a recessive inheritance, meaning that is a very rare mutation that both father and mother are carrier, are heterozygote, and the child is a homozygote. We sent the second brother that was diagnosed later to this lab, and surprisingly, it is also a homozygote to this variant. So it increased the confidence, our confidence in this variant in this family. A third interesting example is the compound heterozygotes. We have 2 of them in our samples. This is one of them in the ANK3, For people who don't know what is a compound heterozygote, it means the father is a heterozygote in one locus in the gene, the mother is a heterozygote in the second locus in this gene, and the child inherited both of these heterozygote variation, and actually both of the copy of this gene are damaged in the child, as you can see here. So the consequence of this compound heterozygote is basically similar to a recessive inheritance. So our future plans is first to extend the search for likely pathogenic variants in other genes, not only in those that were identified in autism or are the top genes in autism. We want to extend this list of search. We want to try to identify pathogenic copy number variations using this exome data. And now we are trying to figure out what are the phenotypes that are correlated with different genes. Maybe we use Joe's classifications of these genes to those that are functional or those that are neurological, and try to find if we see some variation in the phenotypes, because as you saw, we have a long list of phenotypes that we are collected on these children. And we are continue to extend, or we continue all the time to collect samples from these children. And if you have some children or you are interested in collaborating with us with this genetic study, you are welcome to approach me in the break, and we'll talk about it.